This is the narration that goes along with Biology 2E, Chapter 12 on Mendel's experiments and heredity. So early ideas on inheritance showed a little person inside the egg or the sperm, and of course this is incorrect. So this is Gregor Mendel. He lived in Austria at the same time Charles Darwin was alive in the 1800s. Darwin never saw his work, otherwise he would have had a mechanism for evolution to occur. He grew up on a farm in Austria, gardening and studying beekeeping. He entered the Augustinian monastery to become a priest and a scientist. It wasn't a conflict in those days. He studied at the University of Vienna, and he returned to the abbey to study heredity in the abbey gardens, and eventually became the head of the abbey and did administrative work and no longer did science. Mendel used the common garden pea. It turns out peas are a diploid organism, just like humans. So if you understand pea genetics, you understand human genetics. He looked at a number of different characters or inherited features in peas, and he only picked characters that were present as two traits, two varied forms. So pea flowers were either purple or white. If the character was found having more than two traits, he said, oh, that's too complicated. I'm not going to study that particular character. If you haven't had Mendelian genetics before, there's a lot of notation. It's not complex, but there's sort of a learning curve. P is the parent generation. F1 is the first offspring or first filial generation. F2 will be the second filial generation. True breeding plants, he chose true breeding parents. Those are plants that if they had purple flowers, the offspring always produced purple flowers. If they had white flowers, the offspring always produced white flowers. So they were true breeding for the character he wanted to study. And then if you took two true breeding plants that had different traits, a purple flower and a white flower, and crossed them, the offspring were hybrids. They got a copy of one trait from each plant in different forms. Okay, so a trait is a variation. So flower color could be the purple trait or the white trait. We'll go through Mendel's experiments a number of times. He did the same experiment with a bunch of characters, and always picked characters that were present as two traits. In this case, they're showing you the flower color characteristic. And the two traits for that flower color characteristic are purple flowers, well, violet flowers it says here, and white flowers. So he crossed true breeding parents, and he got F1 offspring. The F1 offspring had all purple flowers. That was pretty interesting in itself. Somehow the purple trait covered up the white trait. But he even went a step further, and he said, well, let me cross the F1 offspring. And when he crossed the F1 offspring, the F2 offspring got some white plants back. So even though the white trait was covered up in the hybrid generation, the F1 generation, it reappeared in the F2 generation. So it's as if you had blue eyes that your parents didn't have, but maybe one of your grandparents had. Okay. The purple trait was found in 3/4 of the F2 generation, and the white trait in about 1/4. Okay, so here's just what we just saw, but just in a more dramatic fashion. We crossed true breeding purple and white parents. And here's a purple flowered picture. Here's a white flowered picture. He got the F1 offspring. The F1 are called hybrids because they have a purple trait and a white trait, and the purple trait is covering up the white trait. And then in the F2 generation, when you cross the F1 plants together, you got 1, 2, 3 purple flowered plants for every 1 white flowered plant. So this just explains what we just looked at, that in the F1 hybrid, it's called a hybrid because it has 1 purple gene and 1 white gene. The purple trait covered up the white trait. And he called the trait that covered things up the dominant trait. In this case, it's purple. He called the trait that was covered up the recessive trait. In this case, it's white. But the F2 offspring showed 1/4 white plants. So that shows that in the purple hybrids, the white trait is still there, it's just being masked by the purple trait. We now know the particles causing this are the genes. Mendel didn't know what these particles were, but today we say they're genes, and genes are segments of DNA located on the chromosomes that code for proteins. Okay, more words. So alternate forms of a gene are called alleles. In this case, there's an allele for purple flowers and an allele for white flowers. If the allele is dominant, it covers up the other allele. If the allele is recessive, it's covered up. Usually we take the first letter of the dominant allele, in this case purple, we make it a capital letter, and so we say the dominant allele is purple. We take that same letter and we put it in lowercase. So the white flower would be represented by a lowercase p. And finally, phenotype versus genotype. The trait we see is the physical trait, or the phenotype. So are the flowers purple? Are they white? Is somebody's eyes brown or blue? Are you tall or short? That's your phenotype. Your genotype is represented by a combination of two letters that says this is your gene on the chromosome. These are the forms of the alleles. So you have two purple traits if you're a purple true breeding parent. You have two white alleles. You have two copies of the white trait. If you're a white true breeding parent, and when you cross them and get the F1, you have one of each. You have a purple allele and a white allele. Okay, so let's come back and explain that in terms of the genes. So every individual has two copies of every gene, one from mom, one from dad. So that's what these two letters represent, the two genes on the homologous chromosomes. Okay, the true breeding purple parent had two purple genes. The true breeding white parents had two copies of the white gene. And when meiosis happened, only one little p got passed along in the egg. Only one little p got passed along in the sperm. And all the offspring have a purple form of the gene and a white form of the gene. When he crossed these individuals, you got back 3 purple and 1 white, but you can see that the white plants are all little p, but some of the purple plants are big P, and some are big P, little p. We're going to talk about how to solve that. Okay, when the alleles are in the same form, big P, or little p, it's called homozygous. When the alleles are in different forms, it's called heterozygous. And we can go a step further and say when they're in the same form, and the form is a dominant form, big P is homozygous dominant, little p is homozygous recessive. Please note when you say heterozygous, you never use the word dominant or recessive because it already implies you have one of each. All right, let's go ahead and show how they figured out the F2 generation. So let's start by doing a Punnett square of the parent generation. So when you cross a true breeding purple parent, big P, with a true breeding white parent, little p, and you can read this later on your own, just go through what I'm showing you, we mimic meiosis. So when sperm form, the big P's separate, and so on the outside of the box, we draw a square box and 2 lines across. On the outside, the single letters represent the sperm. Up here, these single letters from this parent separate and they represent the eggs. Doesn't really matter which side you put the eggs on, which side you put the sperms on. Okay, and then big P across, little p down, big P across, little p down, big P across, little p down, big P across, little p down. Inside the box, it represents fertilization. It represents the zygote, the fertilized egg in the next generation. And in the case of the F1 generation, they're all going to be big P, little p. That's the genotype, that's the genetics. And they're all going to be purple. That's the phenotype. Then Mendel took the F1 generation that was all heterozygous, big P, little p, and he crossed them with one another. You can go ahead and do that cross, see what you get, fill in the phenotypic ratio, what did all four look like, fill in the genotypic ratio, what were the genetics. Okay, once you've done that, the answer is on the next slide. Okay, and so that explains where they got the actual ratios. So big P across, big P down, big P across, little p down, little p across, big P down, little p across, little p down. Okay, purple, 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 white. 3 purple to 1 white is the phenotypic ratio. It's what the offspring will look like. 1 homozygous dominant, or big P, big P. You can combine these two. It doesn't really matter whether the little p is first or second. When we write them, we generally write them with the dominant letter first. So we have 2 that have a big P and little p, and then we have 1 that has a little p, little p. Okay? So 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Okay, alleles are different versions of a gene. So the flower color characteristic has 2 alleles, a purple allele and a white allele. Homozygous dominant says they're both in the dominant form, big P, big P. Homozygous recessive says they're both the recessive form, little p, little p. And with heterozygous, you never follow it with a word. All right. So it should be obvious to you, based on what we've done with meiosis, that these genes are on chromosomes. So the chromosomes have between a few hundred genes to a few thousand genes. In the human chromosome, there are 20,000 genes total. The only chromosome that has less than that is a Y chromosome. The Y chromosome has about 40 or 50 genes. It's a little shrimp. But most genes have a few hundred to a few thousand chromosomes. So at this particular location on the chromosome, there's a gene that codes for something. We'll just give it the letter B and say it's in a dominant form and a recessive form. Okay? How do these separate out? It's because the homologous chromosomes will separate during meiosis 1, during anaphase 1. So every sperm or every egg, depending on these, whether this is happening in the ovaries or the testes, are going to have 1 copy of this chromosome and 1 copy of this gene. Okay, Mendel's law of dominance. So this is what we saw just stated more formally. In a heterozygous individual, 1 trait conceals the presence of the second trait. Okay, so both traits are really there in the genotype. So the organism is big P, little p, but only the dominant trait is observed in the phenotype. So with big P, little p, the flower is purple. You don't really know if you just find it in the field, whether it's big P, little p, or big P, big P. The recessive trait will be present in the DNA in a heterozygous individual, but it's not seen phenotypically. That's his law of dominance. Mendel's law of segregation. So this is just based on meiosis, that parents carry 2 hereditary particles, genes. We have 2 copies of every gene, 1 from mom, 1 from dad, carried on homologous chromosomes. But only 1 particle is passed on to the offspring because particles separate in gamete formation. So segregation just means separate. And we now know that meiosis 1 explains the law of segregation. That's when homologous chromosomes come together and then they separate. All right, here's just another example. We won't go through it in detail, but you can't generally predict which trait is the dominant trait, but you just do it empirically. You breed plants together and you see. So it's sort of interesting that within the peas, yellow is dominant to green. So when you breed plants together that have yellow pea seeds with green pea seeds, yellow is the dominant trait. So it's seen in the hybrid plants or the heterozygotes. When you do the F1 cross, you get what we just saw, 3 yellow to 1 green in the phenotype. And so 3 yellow to 1 green in the phenotype, but a genotypic ratio of 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Okay. Sometimes Mendel's experiment is called a monohybrid cross because he's crossing parents that are purebred or homozygous, having 2 different traits, and they're producing hybrid offspring. He did the same for a number of other characteristics besides flower color, flower position, plant height, seed texture, seed color, pod texture, pod color. In every case, the F1 offspring showed what trait was dominant. In every case, the F2 offspring had a 3-to-1 ratio. One thing interesting, I said you can't predict what trait's going to be dominant. For the seed color we just looked at, yellow was dominant, but for the pea pod color, green was dominant. And sometimes after the fact, scientists can explain why, but at the time, that's all he observed. It depends on the proteins that are produced by the genes and how those proteins interact with one another. So our traits are caused by the proteins that are produced, and it just depends what proteins are produced and how those proteins interact, whether a trait is dominant or recessive. Okay, why did Mendel get this feature where one trait was dominant and one trait was recessive? Because he said, you know, this is a simple experiment. I'm just going to pick simple characteristics where the traits are only present in 2 forms. So if a trait was present in more than 2 forms, he said, oh, that's too complicated. It has straight leaves and bent leaves and curly leaves. I'm not going to look at that. So all the characteristics he picked, he found that the traits were present in 2 forms. And so that's why I got this situation where one form was dominant and one form was recessive. So there are a lot of human traits that follow Mendelian inheritance patterns. So we'll talk about a couple of them. One of them is achondroplasia. It causes a specific type of dwarfism, and it's a dominant trait, so you only need 1 copy of the allele to have the disorder. Another one that's interesting that we won't talk about is widow's peak. Widow's peak means you have a little V if you pull your hair back and look at your hairline. What's interesting is that widow's peak is a dominant trait, and yet it's less common in the human population. So simply because a trait is dominant doesn't mean it's very common. Recessive traits, so you need 2 copies of the allele to have the trait. Albinism is lacking the pigment melanin, so it causes a lot of serious problems. And a second recessive trait that causes a disorder is sickle cell anemia. So it's a faulty amino acid within the hemoglobin. So the hemoglobin is a protein that carries the red blood cells. So you need 2 faulty copies of the hemoglobin to have sickle cell anemia. All right, so let's look at the disorder we just mentioned called achondroplasia. So here's the problem. A person with achondroplasia dwarfism marries a person of normal height. What is the chance their offspring will have achondroplasia? Okay, so capital D, we'll use that to mean you have dwarfism. It's a dominant disorder. Little d, normal height. Notice what we just said, that just because something is dominant doesn't mean it's more common. How many achondroplagic dwarfs do you happen to know? I've seen people who have achondroplasia on the little people. The mother on that show is achondroplagic, but I've never known anybody in person, so it's a very rare disorder. Okay, and then they marry a person of normal height, so you'd set it up as big D little d by little d little d. Now to explain that, having 2 copies of the big D allele is a lethal mutation, so you never even get a live birth. The fertilized egg misforms early in the development of the embryo, and so anybody with achondroplagic dwarfism is big D little d. Okay? Okay, and someone of normal height must be little d, since even 1 copy of the big D allele leads to dwarfism. So then the dwarfs big D little d separate in sperm formation. The other parents, little d, separate in egg formation. This is a real person who is an orthopedic surgeon, and he works with other people with dwarfism, and he's very famous. And if you click on this, you'll get the story of how he got into medical school and orthopedic school. And he got in before the ADA, before the Americans with Disability Act. And a lot of schools said, oh, we don't want to take you because nobody will respect you as a doctor because you have dwarfism. And so it was very hard. There was a lot of prejudice against him. Okay, and so you'll see if you do the cross that there's a 50-50 chance that his offspring will be married, that his offspring will have dwarfism. And in the real video that you see, you'll see that he gets married and has offspring. And so you can play it and see what happens. All right, now let's do a recessive order. Let's do albinism. So albinism prevents any melanin from being built. So even people who have fair skin, who are redheads and Caucasian, produce melanin. But the lack of melanin completely is caused by an enzyme disorder, and you need 2 copies of a faulty enzyme to produce no melanin at all. So capital M will be melanin produced. Little m will be no melanin or being an albino. So most of us don't carry this allele. It's quite rare. So most of us will be big M, big M. Okay? If you have big M, little m, you're heterozygous. You're called a carrier. Would you know you're a carrier? Unlikely, probably not, unless somebody in your family has albinism. Then you say, well, maybe I'm a carrier. Okay, and then 2 copies of the faulty gene cause somebody to be albino. Okay, so you could see that there's an enzyme that makes a protein that helps produce melanin, and if you have 1 copy of that gene, you produce enough of the enzyme that nobody even realizes you have the faulty gene. Okay, it's like if 2 doors in the room don't work, as long as people are going in and out through the door that works, nobody even notices. But if neither door works, then you're in trouble. Okay, so the question is, if 2 carriers mate, what are the chances that a baby will be born with albinism? So albinism happens to be a disorder where people with it are fertile and can have offspring, but oftentimes these serious recessive disorders are so serious that people are unable to reproduce. Either they die young or they're very sickly. But in any case, usually for these recessive disorders, people show carriers reproducing, and that's what we'll do here. What are the odds that carrier parents will have an albino baby? What are the odds that carrier parents will have an offspring that is a carrier? Okay, so do the cross between carrier parents. They both have the normal phenotype. It's a recessive disorder. They may not even know that they carry the faulty allele. It may have been passed down for many generations with nobody mating with somebody else who was a carrier. And so oftentimes you don't know you carry these faulty alleles. So the big M and little m separate in the sperm, the big M and little m separate in the eggs. They come back together, big M, big M, big M, little m, little m, big M, little m, little m. It's just like the F1 cross between two heterozygous individuals. Okay? And so what are the odds carrier parents will have an albino baby? 1 in 4. Only the individual that's homozygous recessive is going to have albinism. Okay. What are the odds carrier parents will have an offspring that is a carrier? 1, 2. So 2 in 4. But if you asked a different question, if you said, what are the chances, these are two sisters, what are the chances that this young lady is a carrier? You'd say, well, she's not albino, let's cross this one out. So there are 1, 2 out of 3. There's a 2/3 chance she's a carrier. There's a 1/3 chance that she's not. So if there's a way to test it, I don't know if there is with albinism, she might want to get tested. And that's the answer that we just talked about on that other page, that the odds that the carrier parents will have an albino baby is 1/4. And then the odds that the carrier parents will have a baby that's a carrier is 1/2. Okay, and this is what I just told you, that recessive alleles can be passed down for many generations without people knowing that they're carriers. Okay, so recessive disorders tend to be, or serious recessive disorders tend to be more common than serious dominant disorders because they get passed along for a long time without any symptoms or any ill effect. Okay, serious dominant disorders, they tend to be less common because if somebody has the allele, they have the disorder, and if it's a serious enough disorder, then it's often hard for them to reproduce, and it's hard for them to find a mate, it's hard for them to do the kind of things you need to survive in society, hold a job, and so on. It depends on the disorder and so on. So some disorders, you can do a test to see if you're a carrier, and it turns out sickle cell anemia is one of them. If you have the carrier, you're heterozygous for faulty hemoglobin, they can tell by looking at your blood. Okay? If a recessive disorder runs in your family, you can sometimes figure out your genotype doing a family pedigree. So we're going to do a family pedigree for alkaptonuria, black urine disease. Okay, so there are some conventions that are always true of these pedigrees. Squares always represent males. So men are squares, they're very nerdy, that's how I remember it. Circles represent females. Sometimes the affected individuals are colored in, sometimes they're white. So you kind of have to look at the legend. And then they give you some other things you can use as well. Okay, so these two individuals mate, and they produce 1, 2, 3 offspring. So what it's telling you is that this individual is affected by the disorder. This individual doesn't have it. Okay? And their offspring, one of the 3 offsprings has a disorder, 2 don't. This is a male, this is a female. We won't worry about the diamond. Okay, so here's your problem. This is showing first generation, second generation, third generation, fourth generation. Okay, the blue are alkaptonuria, a recessive disorder. And they say, what's number 1? What's number 2? What's number 3? So based on the offspring of the parents, can you figure out their genetics? Okay, I'll give you a minute, and then we'll look at the next slide and explain the answer. Okay, so all the blue boxes have the disorder. It's a homozygous recessive disorder. So all the blue boxes are little a, little a. That's the only possibility. So box number 1 is little a, little a. Okay. Second question is, how about parent number 2? Okay, so the mom has an offspring with the disorder. The father gave a little a, and since she had an offspring with the disorder, she has to be big A, little a. She's big A because she doesn't have the disorder, but she's little a because she produced an offspring with the disorder. Okay? Over here, the exact same reasoning. These two individuals have an offspring with the disorder, and so this parent must be big A, little a. Now, one last thing, this person has a big A question mark. What does that mean? That means this person, based on their parents, they know they have a little a, okay? And based on her phenotype, she knows she's big A, little a. This person, they don't show the parents, but probably the same thing based on her parents. She knows she's big A, little, he knows he's big A, little a, okay? So in this case, based on the phenotype, they know this offspring is big A, big A. This offspring doesn't have the disorder, but because they know their genotypes, they know this person could either be big A, or big A, little a. And so if you don't know if it could be either, you put big A, because that's based on their phenotype, and you put a question mark for the second one. Okay, so sometimes pedigrees can tell you what your genetics is, and sometimes pedigrees can leave it open to doubt. Okay, so if everything followed Mendelian genetics, the world would be a pretty boring place. So because Mendel was the first one to understand the particulate nature of inheritance, that it's on some sort of particles we now call genes, and these genes get passed along, one of 2 copies from each parent, and then recombine. Usually, most biology courses start genetics, understanding inheritance, with the study of Mendel. But the world would be a pretty boring place if Mendelian genetics applied to everything. So Mendel suggested that 2 alleles exist for every gene, and that dominant genes cover up recessive genes. This is not always true. All right, so consider snapdragons. This is called incomplete dominance, and in real life, this is far more common than complete dominance. Okay, so when you cross a red flowered parent with a white flowered parent, you get pink offspring. These don't look all that pink, but I guess if you saw the red parent, you'd know they're pink. And so it's like a blending of the traits in the parents. Neither the red trait or the white trait is dominant. So the alleles aren't written with capital and lowercase letters. Instead, they're written with superscripts. So the color characteristic to the red gives a red allele. The color characteristic of the white is a white allele. Okay? There's no pink allele at all. Instead, if you're heterozygous, CRCW, the 2 colors blend together and yield pink. Okay, so that would be the F1. The F1 would be all pink. What's the phenotypic and genotypic ratio from crossing 2 pink snapdragons? So do the CR to CW by CR-CW cross. Take a minute, and then when you're finished, the answer's on the next slide. Okay, so the parent cross of a red and white snapdragon produces all pink offspring, and then the CR and CW separate in the sperm, and they separate in the eggs, and they come back together in fertilization. CR across, CR down, CR across, C to the W down. CW across, CR down, CW across, CW down. So now the offspring in the F2 generation are 1 red, 2 pink, to 1 white, and the genetics of the F2 generation are 1 homozygous for the red, C to the R, C to the R, 1 homozygous for the white, C to the W, C to the W, and 2 that are heterozygous, C to the R, C to the W. Okay, and so I put it here. Notice we're not saying that that either the red or white are dominant, because they're not. We just say they're homozygous for the red and homozygous for the white. Okay, so this is actually an easier problem than complete dominance, because the genotypic ratio is exactly the same as the phenotypic ratio. The heterozygous plants have their own unique phenotype. Okay? So, think about if you poured red paint on a white t-shirt and it made the t-shirt red. That's like complete dominance. But let's say the white t-shirt had white paint on it and it made it pink. That's incomplete dominance. So the color we see, or the characteristic we see, depends on what the enzyme products are and how those proteins interact. So you can't predict it. You just have to look at the F1 offspring, and based on the F1 offspring, you can say, aha, it's complete dominance or incomplete dominance. And we sort of know this intuitively, that incomplete dominance is pretty common. So if you have a parent with curly hair and straight hair, it's likely the offspring is going to get wavy hair, intermediate. If you have a tall parent and a short parent, it's likely the offspring will be medium height. If you have a dark-skinned parent and a light-skinned parent, it's likely the offspring will be intermediate in color, darker than the light-skinned parent, but lighter than the dark-skinned parent. Okay, the protein products are blending together, but the genes are unchanged. So the curly-haired offspring could give the wavy hair gene to its next generation, and his kid might have curlier hair than him, depending on the mother's gene as well. Okay, here's a situation where there's a single gene on a chromosome. We get two forms of it, but instead of being present in only two alleles, it's present as 4 alleles. So this leads to rabbit coat color. The 4 alleles are capital C, that's the brown fur color, and it's a capital letter because that's dominant. No matter what the second allele is, if you have a capital C, you have brown fur. Okay? The others are all recessive to the brown fur. The gray is called the chinchilla coat. The Himalayan has a black nose and black paws. The albino has the white fur. And it turns out that if you get the chinchilla, chinchilla blends with both, I'm not sure the chinchilla. The chinchilla blends with both the Himalayan and the albino. So if this rabbit mates with either this one or this one, then the offspring will look intermediate. But with the Himalayan is dominant to the albino. So if these two mate, then the offspring will look like a Himalayan. Okay, why is that? You would have to understand what the protein products are and how those protein products interact. Okay? So it's called multiple alleles because it's still caused by a single gene on a chromosome. Everybody gets 2 copies of that gene, but within the population, 4 different alleles, 4 different forms exist. And that's pretty common that more than 2 forms exist. So if Meta looked at coat color, he'd say, oh, this is too complicated, I'm not going to study it. And it is complicated. So it should be obvious to you, although we didn't talk about it explicitly, but most of our traits are very complex. So whether you're athletic, whether you're musical, whether you're intelligent, those kind of things aren't caused by one gene. They're caused by a combination of gene products and how they all interact. So here's a fellow playing a musical instrument on the streets of India, and being a good musician requires a good ear, it requires good coordination, it requires a number of different skills, and so there's many different genes that will combine to make that in him. Now, oftentimes people who are musical may wind up meeting and marrying somebody else that's musical, and then they'll give music lessons to their kids. So you'll say, oh, that's a musical family, but it's a combination of genes and their environment as well. So let's not forget about environment.